Thanks everybody for joining us. This is a series of webinars that is supported by the USDA, the National Institute of Food and Agriculture within the USDA. And you see the logos here on the screen of all the collaborators that are part of this project, the Upper Midwest Collaborative Plant Breeding Network. This is the third of a six-part series. The first two webinars happened in the last couple weeks, and they're going to happen weekly for the next few weeks. We have already covered goal setting and breeding project design and selecting breeding materials for your breeding projects. Today, we're going to be talking about management considerations for seed quality with an emphasis on seed-borne diseases, seed testing, and sanitation with an emphasis on the upper Midwest region. And so our first presenter is going to be Dan. He's going to be giving some background and overview information on seed-borne diseases. So we can have Dan start to get his slides ready, and then we will have a few other short presentations after that.
Well, I'm really pleased to be a part of this group again. I've done several things with eOrganic, so I'm really honored to be here. So what I'd like to do today is to give you kind of a basic primer of seed-borne diseases of tomato and talk about that, and what I hope that after this you'll have kind of an idea how this happens, how it can happen, and kind of what to do about it. So go ahead, Alice. So the first thing I wanted to talk about is talking about any kind of seed-borne disease or diseases of tomato. There's basically two ways that a pathogen, a microbe that gets into the seed, how does that happen? Well, one is the pathogen must directly contact the seed, and the other is a pathogen systemically enters the seed. And the two oh, go ahead, Alice. I'm sorry. I think I'm controlling my own computer. So if you look in the picture on the left, this is bacterial spot of tomato. And I'm afraid I have a lot of ugly pictures today, a lot of photos. So if you see the ugly lesions on the tomato there, if I split those open under a microscope, bacteria just gushes out of there. And the same thing happens if it rains, or if you were to split that tomato open yourself and try to get the seed out. So the way the bacteria would get in the seed, it would directly contact that seed. You can imagine if you split that open, threw it in a bucket and mashed it up, the bacteria would contact the seed and probably be on the outside of the seed. In the right-hand photo is fusarium wilt of tomato, and you see how half a leaf is dyed and the other half is yellow. This is typical for fusarium wilt. I do not have a photo of the tomato in the fusarium wilt picture, but if I did, it would look perfectly healthy. And that's the point here, is that if you looked at the bacterial spot tomato, nobody in their right mind would purposely save a seed from that. But you could, if you had a little bit of fusarium wilt in it, you would look at that tomato and think that looks okay. So it's important to know that even if you have a good looking tomato, it's possible that if it may have a disease where in the case of fusarium wilt, the microbe, the fungus, goes directly into the seed internally. So in this photo, you can imagine you split open this tomato and it's got lesions on the outside. This is actually bacterial canker, and it's easy to imagine that the bacteria would splash right from the lesion there to the tomato. This is a little bit of a trick question, because actually bacterial canker can be inside or the seed or outside the seed. Go ahead. So what we have in this slide, then, you see the little fake spores that I drew at the bottom of the slide. This is fusarium. It could go right through the root hair, right into the vascular system, and right into the tomato. So in this case, the tomato fruit would look fine, but there could be spores or bacteria in the seed. So does it matter how the pathogen enters the seed? So good sanitation can help to avoid contact pathogens. If I would advise never to take seed from a diseased-looking tomato, but even with a healthy tomato, if you had good sanitation and sanitized the outside of the fruit ahead of time, that would help you avoid contact pathogens. But systemic pathogens may enter a healthy looking tomato seed. So if you're asking me, I would say if you have a field and you're harvesting diseased, or you're harvesting seed from that field, if you had any seed-borne disease in the tomatoes, I would advise you not to harvest seed from that field. However, if you have a large field and you're really counting on harvesting seed from that field, I would say certainly do not harvest from a plant with a seed-borne disease. The fruit should look healthy for contact pathogens, but also if there is any kind of pathogen on any other part of the plant from a systemic pathogen, I would advise you not to harvest seed from that plant. And again, if possible, I would clean and sanitize the outside of that fruit. And we'll talk about seed treatment. So I don't want to spend a lot of time on that, but either heat treatments or chlorine treatments. I don't know if other people talk about that, but that's certainly a way to help minimize the pathogen in the seed. Go ahead. Right here in this photo, this is anthracnose of tomato. And this is an example of, again, a pathogen on the outside with lesion outside. You can imagine if you split that open, how the fungus on the outside could contact the seeds and contaminate the seed. So what I've done here is listed some of the common bacteria or common diseases of the Midwest, and whether they're contact or systemic. So bacterial spot, as we spoke about, will be it gets into the seed through contact, mainly on the outside of the seed. The same thing with bacterial speck. Bacterial canker can be systemic and go in the seed that way, or it can contact the outside of the seed. Anthracnose is a contact. It has to contact the seed on the outside. And then Fusarium wilt and verticillium are both systemic. So they'll get into the vascular system and go right into the seed, kind of bypassing the outside of the fruit. When I talk to people about seed-borne diseases, the assumption is that every seed in that seed lot is contaminated. But usually, seed-borne diseases are inefficient. So usually, you're talking 2% or less of the seed lot has the pathogen in the seed. Well, that's good news. But when we're raising transplants in a greenhouse, and you see the photo there, they're all close together, they're all wet, they're watered from overhead, the disease can spread very quickly. So even if it's only 2%, it can be much higher than that and spread in the greenhouse very quickly. So I wanted to talk about transplant treatments. I recommend applying products every 5 to 7 days. Start at the first true leaf when the tomato transplant has first true leaf. Make sure products are labeled for the greenhouse. Consult the label. I recommend the Midwest Vegetable Production Guide for states that are in that. I think Wisconsin has a different guide. But if you go to nwvegguide.org, you can get into that, and it's searchable. Alternate modes of action. Check for fungicide resistance, and I can help with that. Other plant pathologists can help with that. And then copper resistance is common in bacteria, so you need to check on that. And then I just wanted to mention backpack sprayers versus the common garden sprayers that people use. Sanitation, clean and sanitize equipment surfaces, and then 95% of it is to clean, and you have a picture of dirty trays there or steaks or whatever. Just clean those. If you can sanitize that's great afterwards, and I've listed some sanitizing products there later. But if you sanitize and you don't clean those ahead of time, then you can't. The dirty surfaces doesn't allow you to really sanitize it well. So sanitizing products include bleach, quaternary ammonium, and I've listed some trade names there. Hydrogen dioxide or really hydrogen peroxide, including oxidated xylitol. Oxidate includes hydrogen peroxide and peroxyacetic acid as well in different concentrations. And then chlorine dioxide is another sanitizing product. So seed treatments I mentioned. Sally Miller had a good publication in 2005. Emily Gatch had a good publication in 2016. So if you want to go that route, you can. These publications describe either hot water or chlorine. Chlorine is good for the outside of the seed. Hot water is good to sanitize the outside and the inside, but you have to be very careful not to hurt the germ. Also, if you're going ahead and if they've been treated with fungicide ahead of time, that's problematic. So I recommend separating seed lots and varieties. So if you are growing different seed lots or varieties, I recommend growing them physically different. So that if you do have, for example, if you have several varieties of tomato you're growing, don't grow them right next to each other. Grow them on separate benches. Grow them far enough apart that you can't get a disease to splash from one to the other. And I've seen that happen where it starts in one variety and spreads across all your varieties. So foliar diseases often spread via water. Most transplant greenhouses are watered overhead. And how far? What's the distance? Well, that's as far as water will splash. Know which diseases are seed-borne or transplant-borne, and we've talked about some of that. And again, usually they're contaminated in small amounts, but the disease can spread fast and in a transplant greenhouse. And be careful about saving your own seed. Yeah, go ahead. Go ahead. That was a picture of bacterial canker, and I guess what I was trying to illustrate is just how it's easy to miss. So this is alternative leaf spot on broccoli, and I know we're talking about tomatoes today, but I wonder if you can see the difference in the seed lots there. What's happened is in the first greenhouse they put most of the seed lot continued on towards the back of this greenhouse, and I think if you click one more time, yeah, see, you can see there's a different variety there. And this disease, which is alternate leaf spot of broccoli, is going to go right from the one variety to the other. And that's why I'm recommending to separate varieties, whether it's broccoli or whether it's tomatoes. Separate those varieties in distance so you don't get a disease go right from one variety to the other. At least you'd save one of the varieties. So I used to work for a seed company. I know seed companies go to great lengths to sell healthy seed. But when seed is tested, it's usually done by what's called a grow out test. And the way that's done is just simply to grow the plants and the seed in the greenhouse and in a very conducive condition, watered overhead, keep it very high humidity, and try to get disease out of that seed. And if you get diseased seed, then they throw out that seed lot. And what I showed a picture below, and I know this is watermelon, but the concept is the same whether you're testing watermelon or cantaloupe seed. But when you grow out this seed and grow out tests, take a sample of seed in trays in the greenhouse and look for disease using very conducive conditions. When you do that, the seed that you tested is destructive. Okay, you grew that out, and hopefully you found that that's healthy, but you can't sell that seed because you grew it out. Therefore, you have to sell seed that has not been tested. I hope I'm being clear here. So when seed companies have a seed lot, let's say there's 100,000 seed in there. Let's say they grow 10,000 seed out in greenhouse tests, then they're left with 90,000 seed, and they'll sell that. And they're selling seed that's been tested that from that seed lot, but the actual seed they're testing hasn't been tested because testing it involves destroying it. Okay. So I want you to understand that it's possible to sell seed. The seed lot's been tested, but the actual seed has not been tested. And we can, if you have questions about that, I'm glad to address that. Go ahead. I wanted to leave you with this photo. When I go into either a greenhouse or a field situation, what I like to see is it's well organized and clean and sanitary, not dirty. Kind of like how we would go into a restaurant and you want it to be clean, you want it to be well organized and not cluttered. And the same thing will be true with any kind of agricultural operation, including a transplant greenhouse. So I like this photo. In addition to being just a nice painting, you you can see the where they're eating and the kitchen part all in one, and then and hopefully if when I see a, a greenhouse, I want to think if this was a restaurant, what would I want to eat there? So that's what I try to keep in mind with my operations, and I think it's a good idea for your operation. And I think, Alice, I have one more photo or one more slide that has my contact information and maybe something to remind us of warmer climates. So I think there was some questions. I'll go ahead and try to address those.
Yeah, I can read some of these questions up for you, Dan. Do you know of any resources to know the difference between the systematic versus the contact disease? How could somebody know?
Well, again, for tomato, I tried to address those. I think the bulletin that I gave you, Michael, I think that talks a little bit about those. Um, uh, and then I think some of the other webinars we've had, Alice talks about those. But I tried to address that in that one slide back there, looking at for at least for tomatoes, the contact versus systemics.
Great. Yeah, we'll share that link with everybody. Can Phytophthora infect seed, and can it survive for 6 months or more in the seed?
To my knowledge, there's been no publication that that Phytophthora can be seed-borne. I don't know of any information that suggests that that's true. And if it does become true, I hope I'm retired by then.
So let's say you yeah. Go ahead.
Well, anyway, no. So no, as far as I don't know.
That's good. Let's say you unknowingly harvest seed from a tomato with a systemic disease. Would that disease transfer to 100% of the seed lot?
No. And let me return to the Phytophthora. You're talking about late blight, perhaps.
Late blight. Yep.
Yeah. So that's not. I think that it's listed in the book as being it can be seed-borne. I don't think it's really commonly seed-borne. When you're saying Phytophthora, I was thinking about Capsicum. But yeah, if it is seed-borne, it can last 6 months in the right storage. But I don't think that that's commonly seed-borne as much as the ones I mentioned. What the second question you asked was?
Yeah, whether it would be 100% of the seed lot.
Yeah, and that's be very rare. It's usually just a few percent. You'd have to really try to get it. It's an inefficient process, which, like I say, is good news for us.
This question might be answered by our later presenters, but can steam treatment be used for any of these diseases?
Yeah, I've seen some information that, and I'm not familiar with that, but I have seen there's some steam treatments. Gosh, I think that came out one of our other webinars, Alice, about a firm. Actually, it is a seed company that offers to steam treat your seed, and I can't pull that out of my head right now. But yeah, there are some resources for that I can't remember right now. But steam treatment is an alternative. Yes. High mowing seeds. Thank you.
Yep. And a question here, I'm not sure I totally understand the question, but if a variety is known to be tolerant to some of these diseases, would you potentially save seed from some plants that are showing some level of resistance?
I guess I'd be hesitant to do that. So, I mean, if there's partial resistance in a variety, and yet you have some inkling that disease is in there, it's possible you could get enough disease in there to infect the field or infect the soil for a later date. So, I mean, you're asking me, I'm a plant pathologist, and I'm always going to be cautious, but I'd say no.
And a question about quarantining plants, can it be used for bacteria or viral pathogens or fungus?
So when you talk about quarantining plants, I usually think of quarantining on kind of a large scale. You know, these counties or these states are quarantined and can't send these other places. I don't know if you're talking about quarantining, like if you had, for example, let's say if you had a tomato plant that you knew had a certain disease, but you wanted the seeds and you grew them in a separate field and quarantine them. I don't know if that's what the questioner is meaning. You could do that, but then you risk contaminating the soil in that other field. I'm not sure if I understand.
Great. Thank you, Dan. That's all the questions that we have here for you. Really appreciate you taking the time to join us and
Okay.
present that. So I am going to now present a few slides here. The first thing that I want to address, a big management consideration for seed quality, here I got that, is the climate and the regional environment that you're working in. This was a resource that Organic Seed Alliance published. It was almost 10 years ago now. Still a great resource, highly relevant to what we're doing. This resource has a little bit of a focus on the Pacific Northwest region of the United States. Our group here, our breeding network group, is going to be releasing a similar guide with a focus on the upper Midwest. And actually, we're looking for a few case studies for that report. So if you are in the audience or you're listening to this and you have experience with adapting or mitigating the effects of climate on your seed production, feel free to reach out to us, and we may consider including you as a case study in the upcoming report. But this guide has a lot of great resources, things to consider when you are producing seed. When you are growing plants for vegetables, you may not have the same challenges as when you are producing seed. The environment that you are working in can affect the pollination or the fertilization of your seed, which can affect the quality of the final product that you're getting. The temperature, the length of your days, the length of your season, wind, moisture, precipitation can also affect your ability to produce high quality seed. Another thing that this report features is considerations for field selection, whether you have different fields on your farm that have different conditions, or you have access to protective structures like greenhouses, or you can use row covers, individual small tunnels to protect your seeds. There's also information in this report about the differences between dry seeds and wet seeds. So whether your seeds are coming out of a wet fruit like a tomato, or they are dry seeds like grains and other things. I also wanted to talk about resources that are available for folks to diagnose diseases. I hope that you won't let the fear of seed-borne diseases stop you from your breeding goals. I know a lot of people will have watched the first two webinars and they have got these ambitious breeding goals. They're excited about some of the breeding materials that they're working with. And I just want to let you know that there is help available for you to identify and diagnose some of these diseases. There's a lot of images and descriptions available on the internet that you can use, and also testing kits that you can use to diagnose some of these diseases at home. You can also take samples of the plant tissues and send them to diagnostic labs that can help you identify them. And I would also encourage you to find a local or a regional expert that can work with you to help identify these diseases, and then even once you've identified them, come up with a management plan to help address and mitigate the effects of some of those diseases. So I'm sharing this image here of the National Plant Diagnostic Network. This is a resource that the USDA has cultivated. If you go to that website, you can click on your state or your region, and they will provide you a list of diagnostic labs that you can work with or send materials to. Many of them are university extensions. Some of them are Department of Agriculture. But just know that you're not alone. You don't need to be a plant pathology expert to be a plant breeder. There are a lot of people out there that are professionals in this field and would be happy to help you. One other thing that I'm going to be talking about is sometimes you have really heavy disease pressure on your farm, and this may or may not be a seed-borne disease. It may be a disease that is not transmitted by the disease through a systemic or contact mechanism. And so a lot of plant breeders have actually taken this disease pressure and used it to their advantage when their breeding goals include breeding for disease resistance. So some people refer to this as a disease nursery, and it is almost like what you would do if you were to break all of the rules of disease management. So you are leaving crop debris in the field. It may be really moist. You may have your plants spaced too close together. You may leave some of the fruit from previous crops in the field. And you grow your breeding population out in there after you've identified some good breeding materials, and you screen for disease resistance. And you may identify different levels of resistance. There may be some plants that are completely destroyed and don't produce any seed. You may see some plants with a moderate level of resistance, and you might find a few individuals that have very high levels of resistance. And that could be a start toward breeding a new variety or population that has greater resistance to the disease that is affecting your field. There's also things to consider, whether the mechanism of resistance is just through a single gene or just a few traits, or whether your population is just very vigorous and hardy and has multiple mechanisms for being resistant to those diseases. And that second mechanism is a more robust level of resistance to disease. So next we're going to have Jenn talking about some of the mechanisms for sanitizing seeds, and then Erica is going to come on to talk about steam treatment and some of her experience with managing seed quality.
Okay, hi everyone. My name is Jenn Cava. I am the trial manager for our collaborative plant breeding program at with this group. I'm going to be going over our organic approved seed sanitation procedure that we use here in the lab. Because it's collaborative, we end up sending out seed to a lot of farmers and gardeners and growers. So we make sure to sanitize all the seed we send out so that we're not spreading anything that that might be present in our greenhouses or fields. So this procedure is used in a lab at UW-Madison. Obviously, some of the equipment you might not have, although I think most of it is pretty attainable even if you're not in a lab setting. It's approved by the Midwest Organic Services Association, and we currently use it successfully for tomato and pepper seeds. I don't think we've tried to use it on anything else yet, but we haven't seen any effects on germination in these two crops. I'm going to be going over an overview of what happens in this process. I'm not going to go step by step, but we are going to be able to provide a document outlining everything in a more step-by-step manner if you're interested in using this process. So all of our seeds are put into small muslin drawstring bags that are very permeable, so liquid can get in and out of them easily. And to do the actual treatments, we hang these from a glass stir rod into a beaker with the solutions. The first solution we use is a 10% solution of trisodium phosphate, and the seeds are soaked for about 30 minutes. So in this picture, you can kind of see our setup with, 8-ish seed lots, small seed lots. So these are usually around 300 to 1,000 seeds in these little bags. You can see it's on top of a stir plate. So we use a stir rod while the seeds are soaking to help keep the solution moving around and making sure it's getting in contact with all of the seeds. It is in the picture that's also like a heater. We don't use any heating element. It's all done at room temperature. One important thing to note, TSP, for example, where we are in Dane County, Wisconsin, it can't be just poured down the drain. It's a chemical cleaning agent that you're going to want to check what proper disposal methods are needed for your area. It's also one that does require some extra PPE, including like gloves and goggles, and you don't necessarily want it to sit on your skin for a long period of time. Once those 30 minutes are up, we end up switching to a bleach solution. We basically just pull the a whole stir rod out, and then have another beaker with a 0.5% solution of bleach. Put it on the same thing, put a stir rod, and let that one go for 20 minutes. So bleach it has chlorine in it, so when Dan was mentioning using chlorine to sanitize seeds, that's part of what's happening here. Once they're done being soaked in the bleach, we rinse each of the seed lots separately for at least 5 minutes to get any additional bleach residue off for the most part. And you can see this picture is, we use one of those really fine mesh kitchen strainers, and then just are rinsing the seeds. This is the part that takes about the longest. So you can see between the soaking and the rinsing, this process can take a few hours depending on how many different seed lots you have. We try not to do them in huge batches just because that's a really long time to be standing at the sink rinsing seeds forever and ever and ever. But if you have the time and equipment, it is organic approved, and as far as we can tell and have seen, it works very well to make sure everything is, we're getting rid of those contact seed-borne diseases. So after they're rinsed, we place the seeds on coffee filters to dry them. So in this picture, you can see that there's 5 different seed lots on a drying rack, and we fold them up into quarters just to, there's less area for the seeds to kind of escape in case you drop them while they're still wet. The seed stays in the coffee filters pretty well, and we usually will let them dry for at least 2 to 3 days, and then once that is done, we can get them counted and put into envelopes for storage. I believe that is all I have, and I'll be we're doing the rest of the Q&A after everyone else's talk, so I believe I am passing this off to Erica to talk about other types of sanitation methods.
So I'm just going to briefly talk about a wide range of things, but also as a seed farmer and plant breeder, the things I think about when I'm doing disease control, specifically seed-borne diseases. So first, I just think about what are the skill sets needed to be able to do this work well, and being a keen observer is really important, so that you notice, what's out there. Being a detective to be able to, problem solve and look at different plant specimens and figure out how to get help, and then discernment, being able to make decisions about, do I save seed from those plants or not, or what treatment do I do? Okay, so any farmer knows that scouting in the fields is really important to check for disease. I scout our fields once a week, and obviously just looking for anything abnormal. But here are 3 different examples of 3 different things that you could see on tomatoes, and they're all quite different. So the one on the left is blossom end rot, which is a calcium deficiency, so it's not even a disease, nutritional deficiency. So not seed-borne, not a problem. The one down here in the lower bottom, you guys probably know that, the Phytophthora or late blight, which, as Dan mentioned, is not seed-borne. So even though we may not actually get seeds off of these plants because the plants might be dying too quickly, it is not seed-borne. And then the one in the upper right-hand corner is bacterial speck, which is seed-borne. So just being able to observe and then use skills to figure out what you got. All right, so once you find some sort of symptoms out there in the field, you have to figure out how to identify the disease. And on the left here, we have peppers with alfalfa mosaic virus in them, which is seed-borne. And then on the right here, we have pepper that has iron deficiency. So you can see that, especially with viruses do weird things that are really, really hard to identify. So it's really important to get good identification on your disease at all. And I'll talk about identifying viruses a little bit. So after you've found symptoms on your plant, you want to figure out what is going on. And anybody who's learned about horticulture or done any agriculture training learns about this kind of disease triangle. And in order for disease to happen, you have to have a susceptible plant. And you have to have the pathogen around. So whether it's a virus, a bacteria, or a fungus, and then an environment that's conducive to the pathogen being able to grow. And this can also include, like, conducive environment could include any kind of like insect that could say spread the disease. So as an organic farmer, especially, we need to even look at these things more closely and specifically use these to ask questions. So what kind of questions do we need to ask? And one of them with looking at this is with the alfalfa mosaic virus. The alfalfa mosaic virus. Where does the once you've identified it, where does the pathogen come from? How is it transmitted? So we're looking at, this quadrant of this section of the triangle here. Where is that pathogen coming from? And how is it transmitted? And is it seed-borne? So once you've kind of identified in order to identify the disease, people have mentioned some different resources for doing that. A great resource is often your university extension. So for us, the Michigan State University Extension, they're really great at identifying bacterial diseases and fungal diseases, and I can send them samples. I can actually call them and get quite a bit of free advice, but they really don't know much about and can't really deal with viruses, for example. So that's when I would go to a private lab. I just have Agdia up here as an example. As a private lab that we use to send virus samples to for them to test for viruses. Okay, so we've got alfalfa. So we figured out we have alfalfa mosaic virus in our pepper, and then how once we figured out what virus, what disease we have, how do we know whether it's seed-borne? There's lots of different resources for this. I really like the one in the back of John Navazio's The Organic Seed Grower book. On the right here is just, it's several pages of these charts, and you look it up by the family. So for this example, you can look at onion, and you can look at what the diseases are that are seed-borne. And then he also has his ranking in here for how severe the disease is. So that's a great resource for figuring out, because once it's like you might come up with something like late blight and go, oh, is this seed-borne or not, right? And so then looking up, okay, it's a problem, and that's really important, right? You need to know whether it's seed-borne or not, to whether you're going to save seeds from those plants or whether you're going to use that material for plant breeding, etc. So then another thing to think about, which the disease triangle can help us think about, is how can we use life cycle information about the disease to help make informed decisions about seed-borne diseases. So in this case of alfalfa mosaic virus in peppers, so yes, it is seed-borne, but then we're going to look at that life cycle. So it starts out, it's in a plant. So on my farm, we have some alfalfa fields, and it turns out that aphids will actually, feed off the alfalfa and get the virus in their body, and then come to a pepper plant, and they can inject it into the pepper plant. And then, eventually that pepper would produce fruit down here. There's no picture of pepper, but it can be in the pepper. And then it would be inside the seed. And so that's where the problem happens as a seed-borne disease. That you don't want to sell that seed or breed with that seed. So in this section of the life cycle here, though, what we learn is, by doing research, what I learned was, and talking to people as well, is that this particular species of aphid doesn't travel very far, and is not a very effective spreader of alfalfa mosaic virus, and the virus doesn't stay very long in their bodies. So what I learned on our farm is that as long as my peppers aren't super close to the alfalfa, we often have a lot of cover crops. So we were just sometimes like planting right into hay fields, and so the alfalfa was literally like almost touching the pepper plants. So, we've learned over time, at least, 50 feet away, 100 feet away. And then we really have almost no, alfalfa mosaic virus. So that's an example where doing that research, learning the life cycle is a way to, like, okay, how can I manage this disease in my field and make sure that I don't have it? And then occasionally I'll have like one plant and, hundreds of plants, or 2 plants or something, and making sure I'm scouting the fields, right? And so if I see it in the fields, then I'm going to pull out those plants so nobody accidentally harvests peppers off of them. And as you all know, in organics, prevention is key, right? All right, so once we figured out that something is seed-borne, then as other people mentioned, that I was going to talk a little bit about doing hot water seed treatment. One of the great things about hot water seed treatment, as opposed to some of the things that Jen was talking about, is it is easier to do just on a farm with less equipment, and you don't have to, the TSP is pretty, damaging to the skin. So, you have to, dispose of it special. So the hot water treatment is a pretty easy thing to do. It does take, you have to be very careful doing it. But so the hot water essentially, so you can see by this chart. So here we have, so this chart shows the crops, and then the amount of the temperature that you want the seed exposed to, and then the amount of time that is exposed, and then the diseases that are controlled. So with Brussels sprouts, you would do a hot water treatment in 122 degrees water within 1 degree. You don't want to go too high. It can be between 121 and 123, but no, you don't want any more fluctuations than that. Ideally, it would even be closer to 122. And you're going to do it for exactly 25 minutes. With that, though, you get control of all of these diseases here. And this is just a great resource I found at UMass that actually has quite a few diseases on it and quite a few crops listed, and all these really important details about the time and the temperature, which you have to be, very careful with. You'll notice that. Yeah. Can
you advance a slide? I think we're seeing the table.
Sorry, of course I'm looking at the notes up there, and farmers and our ability or lack thereof with technology. Okay, so yeah, sorry. So here's this slide. Thank you, Mike Cole. This slide. So here's what I was talking about: Brussels sprouts, the temperature of 122, and then the time, 25 minutes, and then here are all the diseases controlled. And you can see this. It's located at UMass Extension, and the link is down there, that you can be able to find this yourself. So this is a great way to look up what diseases you can do with hot water treatments, and which crops, and the details. When you're doing those, that hot water treatment, you need to think about a few things. First of all, you can very easily kill seeds, right? If your water is too hot, you could kill seeds really easily. So you want to be really, really careful and do that really mindfully. So one question that I often ask myself is, when do I want to do the hot water treatment? I'll talk a little bit more about when do I want to do the hot water treatment? Of course, there's Kiwi when she was a puppy, and this, the cutest thing in this picture. But what I was just showing is that we do grow a ton of tomatoes. And so I have all my breeding lines, and we do tons of trialing, and we have, we get seeds from seed swaps and all over the place. And so what we do, and which is similar, the same thing that what Jenn does is they, treating the seeds. I do hot water treatment on a lot of the seeds before I plant them in the field, just to make sure that I'm eliminating those diseases. So that's the best time to do it. Doing hot water treatments can shorten the life of the seed viability, and the germination rates. So sometimes it is done on seeds before they are sold, but typically it's a better practice to do it on the seeds before they're planted. And it's also a lot easier because you're doing much smaller batches that way. So the best resource that I have seen for doing the hot water treatment is this one that OSA was part of. It's on there, or no, maybe it wasn't OSA. Actually, it's somebody at Oregon State University. It's a PowerPoint that you can find online, and there's a resource here, and it shows you step by step how to do the hot water treatment. So you must be really accurate, as I mentioned, when you do it. And they show you some different equipment. Yeah, I have that in the next slide, so I'll tell you that in a minute. But you want to do it carefully. And the first time you do it, do a test batch. Take some random seed you don't care about and do that test on it first, and make sure you have your systems in order and you have really good notes, and you have your alarm with you, and you have everything you need to be able to get it exactly right, so you don't kill your seed. I've heard of people killing large batches of seed by not learning how to do it properly first. So just a word of caution with that. Yeah, I missed it. Okay. So here's from that resource. And here are some of the equipment that they use to do the hot water treatments. The biggest thing is that you need to maintain the correct temperature of the entire bath. So, obviously if there's heat from the bottom, and there can be the temperature might not be uniform throughout the hot water bath. So it's really important to keep that water moving in there and really monitor that temperature quite well. So here are some different setups that they have. I guess Frank Morton sometimes uses coolers. I've seen some sinks that he has too. And then high mowing. Actually, high mowing I think has moved pretty much to the steam treatment, but they have a whole very expensive setup for that, which if you have questions about it, I know somewhat about it, but I'm not going to talk more details about it. It is more, it's different. So these are just some basic cheap setups that you can do. But one of the things that I figured out was, and I'm sure other people are doing too, is I bought a sous vide that is like this, which is so you open this thing up, and what it does is you can set it at a certain temperature, and it will maintain that temperature and you can put a timer on it. So that's what I like to use, and as long as I don't pack too many seeds in there, it actually circulates the water. I do go and manually stir it. I go and check it every 5 or 10 minutes just to make sure that the temperature is right. And to the right, I'm just showing I have like an instant thermometer as a backup thermometer, because if that sous vide was broken and it was baking the seeds at 150 degrees because the thermometer was broken, and I have a lot of important seeds on there, I've got a backup thermometer to know how hot that is. So yeah, so that's all my presentation, and I think we're gonna go to questions now.
Thank you, Erica. Thank you, Jen, for those presentations. We've got some questions coming in for you. I'll try to kind of organize the questions in an order that makes sense to me, at least. Jen, do you change the solutions between batches when you're sanitizing?
Yes. Normally we're only doing one batch in a day, but if we are doing more than one, then yeah, we'll switch the solution in between. You can actually see especially when you're doing the TSP part, a lot of the junk from the outside of the seed comes off, and it does get dirty. So it's best to get a new batch in if you're going to do more than one in one day.
Sounds good. Do either of you know if it's possible to, or Erica, did you want to say something about that question? No? Okay. Do either of you know, can you sanitize your seeds right after separating the seed from the pulp, like while they're still wet before you dry them, or is it better to dry them first and then sanitize?
At least with our chemical sanitation, we do actually go straight from fermentation of tomato seeds to the little bags and the TSP treatment. But we've also done it where the seed was dried before. So it seems it works either way. As long as you get most of the pulp out. You don't want the pulp in there with them while they're getting sanitized.
Just to chime in quick, I think the important thing that Jen said is that it's after fermentation for the tomato seed. So they're not dried, but they have the gel removed so that the surface sanitation works.
Great point and clarification. Thank you, Julie. Question about a heavy residual chlorine smell after some batches of tomatoes have been sanitized using the organic methods and percentages. Does anybody have any thoughts about that?
At least when I've done the rinsing at the end, I will just honestly, even if it takes more than the 5 minutes, I'll go until I don't smell the chlorine anymore. And if I need to step away and breathe different air and come back and smell it again, that's what I end up doing. The 5 minutes, we picked that because it gets it down to 4 parts per million for the bleach. And I have noticed a difference in how long it takes the smell to go away depending on the size of the seed lot. So the smaller seed lots go faster in terms of getting rid of the smell and lowering the concentration.
great. And everybody can feel free to keep the questions coming if you have them. Another question here, would there ever be a scenario where you would use a hot water treatment and the chemical treatment? They wonder if there are any pathogens that are killed by only one or the other, or is it more or less interchangeable?
Julie, you want to take a stab?
Sure, and I apologize, I don't have a webcam on this machine. So the sanitation protocol only kills surface contact pathogens, so it won't work for systemic pathogens. The seed, hot water, and steam treatments will work for both. And the reason we don't use them is because they take a lot more skill and a lot of, very careful attention. And so it's harder to do them, at least for us, when we have a lot of small batches of breeding seed. I think that Frank Morton has a really good setup for that. And then there are other resources that were mentioned to help set yourself up to do that well. But those two will kill some systemic pathogens. Systemic pathogens are always harder to kill than surface ones. But because we mostly see surface pathogens and it is far easier to get the protocol right, we use the sanitation.
A question about blossom end rot. Erica mentioned that this isn't necessarily a disease. It's a symptom of a nutrient deficiency or, some issue with your soil or water access. Is it possible to do any form of selection for blossom end rot resistance in a breeding program?
I have not done that, but I would suspect yes, because I know there is quite a bit of variability in blossom end rot in tomatoes. So I think even just screening lots of different varieties for blossom and rot and, creating the conditions specifically like Mike Lordon mentioned, a disease nursery, almost like make sure the conditions are there and then screen for it and do replicated trials so you know that you're actually identifying varieties that are more resistant to it. So I would suspect that you can do that. I have not, I don't know specifically though.
This is an interesting question and maybe overly broad for this webinar, but I'll put it out there anyway. The question is about GMO breeding for resistance to viruses or bacteria. Does anybody want to address whether they think that GMO breeding or genetically modifying them is better or worse than classical plant breeding?
I can take a stab in that. I think that classical plant breeding has the capacity, Mike Lordon talked about kind of the horizontal resistance, to have multiple genes or multiple parts of a plant that are actually creating disease resistance. And we know that disease resistance persists better over time. And you can do that with classical plant breeding. You can't really do that with genetic modification.
I'll add just a brief thought about that from my experience learning about evolution and the what some have called an arms race between things like plants and pathogens. And you may spend a lot of time and effort breeding a specific gene for resistance to a pathogen. And it can happen pretty quickly that the pathogen can evolve to become resistant to that one change. So like Erica mentioned, horizontal resistance. There's a book all about horizontal resistance, and that method of resistance, like Erica mentioned, tends to be a little more robust and last longer. So I think you will find most independent breeders at a smaller scale would lean toward classical plant breeding methods. Another question about a multicolored tomato, the Berkeley tie-dye tomato. They noticed that there were multiple colors in that seed. Is there any, does anybody here have any ideas of whether that may have come from a treatment or why there would be a difference in the colors of those tomato seeds?
In the seeds themselves or in the fruit?
On the seeds themselves, I believe they noticed like multicolored seeds or different coloration in the seed.
Sometimes during the fermentation process, you can have molds or other things that make the seeds like dark in color. Other colors than that, I don't know.
I mean, if the seeds are treated with some sort of coating that would be colorful, I could see that. But like, as far as our methods go, I've only ever seen like, slight differences in how dark of a brown tomato seeds are.
Catching up on the chat here, making sure I didn't miss anyone. If anybody else has any other questions, feel free to put them in. It looks like question about whether that resource list will be sent out to attendees. Alice, are we sending out the slides at the end of these presentations?
Well, they're already up on the eOrganic website. Let me just put the link in the chat here. I can try to remember to put them in the follow-up email as well, but they're on that. They're on the page with all of the resources from all of the different webinars. Let me just put the link in the chat here for everybody, and I'll try to remember to put it up in the follow-up email as well. So here's a link to the slides. I just put that in the chat. And then the link to the slides from all the webinars that we've had, as well as the recordings and some additional resources. I just put that in the chat too.
Great. Thank you so much, Alice, for that.
Mm-hmm.
A good comment here from Craig that rust in the water source may also affect the final tomato seed color. So the water source that you're using for soaking, fermenting, rinsing, whether there's certain minerals in that water may have an effect on that final seed color. I'm not seeing any more questions. I'll give it another minute or so just in case anybody hasn't done there. Another comment just came in here. Is that the seed coat may vary in color. That may be related to the seed maturity. I've noticed that as well in some crops. They said in fruit tree seeds, but also in some vegetable seeds that immature seed may have a slightly different color than a fully ripe or mature seed. It's another good point there. If any of our presenters have any final thoughts or things that have come to mind during this discussion and you want to raise those now, would be a good time. Another question came in here about the late blight, and they said in some years there's no infection in their fields, especially in a dry year. They want to know if there's any way that they could store that blight over the winter, or store it for periods of time to use as some sort of inoculant.
Ooh.
It's probably a scary thought for some people, but is that possible?
Technically, you could, like, it stores in things like potatoes, but I'm telling you, any potato and tomato farmer is not going to be happy with you if you're like actively releasing late blight. I don't know about, maybe Julie can answer this, but if there are tests, so one of the things, if you're doing plant breeding and you start a bunch of seeds, you can actually take like a hole punch and get a sample of that leaf and then set it off for genetic testing to test for disease resistance in your in your plants. And so that might be a way of doing it without having to actively encourage late blight, which not only affects you, but neighboring farms.
Yeah, there are a few known genes for late blight resistance. So you can get those tested with molecular markers. Actually, you just send the tissue and then you get back the data, so you don't need to do any of it yourself. You could also do an assay in the greenhouse, or what's called a detached leaf assay, where you take a leaf and put it on a Petri dish and then inoculate that leaf inside and see if it appears to be resistant or not. I don't know if there are protocols developed for Phytophthora. That would be a good question for Dan. I know that in Wisconsin, it's illegal to release it intentionally. Not in other states where it occurs more frequently. So I don't know what your law would be where you are. But because it is such a devastating disease, it's often pretty highly regulated.
And where could you send a sample for that genetic testing if you know?
It depends somewhat on which country you're in. There's a few in the U.S. and we could certainly provide links to those. We could make a little handout and provide some links to those. There's 2 or 3 that do pretty routine samples. I would have to look which ones offer the late blight testing. And I don't know about the Netherlands. I guess the question came from the Netherlands. So gotcha. I would contact probably a local university or a seed dealer, someone that sells seed potatoes. They may know what the regulations are.
Gotcha. There's a question about like good biology, inoculating seeds with potentially beneficial microbes. I wanted to point out the first resource in that list of resources there, that YouTube link. It was a presentation from the Seed Growers Conference, Organic Seed Growers Conference in 2018, featured Dan as well as Jim Myers and Lori Hoagland, I believe. And they presented a topic that has a lot of similarities with what we're talking about today, a lot of the seed-borne diseases, managing, diagnosing. They also talked about some of upcoming research or newer research that is looking at other biology, other microbiology on seed and within the seed, and looking for some potential benefits of other microorganisms. A lot of times we think of microorganisms as germs, but a lot of us are recognizing more and more that there are a lot of beneficial microorganisms. So does any of our presenters know anything more about this topic of seed inoculants after the seeds have been sanitized to sort of reintroduce a healthy microbiome?
I've learned a little bit about mycorrhizal fungi, and I actually don't know the details. I think there are some certain types of mycorrhizal fungi that you can try to inoculate with. I don't know what the data's been out there. I think some of it's been more anecdotal with organic farmers.
I think there are a couple of companies that are developing products, but mostly to prevent things like damping off and not necessarily to treat seed-borne diseases. I think it's an area where there is a lot more research that should be done, because it could potentially be, especially for surface pathogens, it could potentially work well if they just outcompeted those pathogens. There's certainly research that's been done on trees and other things to use beneficial microbes, but I don't know any treatments that are currently in use on seeds for this type of application. It doesn't mean there aren't any, just I'm not familiar with them.
Yeah, as a follow-up to this webinar series, we're going to be releasing like a toolkit with additional resources, and I think this is a really good question that maybe I'll do a little more digging in to try to find out what has happened since that 2018 Seed Growers Conference, and see if there's anything new that has come up and that would be practical for independent plant breeders, collaborative plant breeders. Well, I'm not seeing any more questions. I don't think I missed anybody, but so I think that might be the end of our webinar for today.